Hello friends, today we are going to discuss about microbiota. I am Dr. Suresh Badadmat, Professor of Psychiatry working at NIMHANS, Bangalore. In this video, I am going to discuss about the definition of gut microbiota, what are the functions of this microbiota, what are the psychiatric illness associated with microbiota, if there is a dysfunction of microbiota, how to be rectified, what are the practical implications, what are the current research and conclusions. All disease begins in the gut, Hippocrates said long back. Let's understand what is microbiota. The human gastrointestinal tract represents one of the largest interface between the host and the environmental factors. If you see the GIT, that is gastrointestinal tract, is hardly 5 to 6 meters. But however, because of the finger-like projections, villi, the surface area increases. The total surface area is approximately around 30 to 40 square meters. However, earlier it was considered to be 300 to 250 square meters. But now the research have showed that it is the surface area of half of the badminton court. Approximately 50 to 60 tons of food passes through this GI tract in a lifetime. That means this food will be digested, fermented, and utilized for energy. Hence, this gastrointestinal tract is considered as a bioreactor. Along with the food, when you consume, there are many threats. It may be bacteria, it may be virus, fungus, also goes through this GI tract. To neutralize this threat, there should be an immunity. That means there should be a defense mechanism that is called as microbiota. So microbiota not only ferments and digests the food, but also acts as a first line of defense against the attackers, that is bacteria and virus. Humans do not have any mechanisms to digest some of the food. So that means we require somebody else to help us in digesting. So these are the helpful bacteria or commensals bacteria which are there in the gut lumen, and they play a very important role in fermentation and digestion of the food. These collection of bacteria, archaea, eukarya, fungi and virus which colonizes the GI tract are termed as gut microbiota. There are 100 trillion microorganisms which are present in the human gut. These organisms have evolved, or I can say co-evolved along with the human beings over thousands of years to form an intricate and mutually beneficial relationship between the host, that is human, and the gut microbiota environment. They are called as commensals. That means the food we take is required for bacteria. They will ferment this and they will utilize part of it for the energy. Whatever it is left behind will be utilized by our human body for fermentation and then the digestion and usage for energy. So let's look into this diagram. You can see the GI tract starts from the mouth and ends at the anus. So here it is around 5 to 6 meter in length, but however, entire gut length, you can see there are 100 trillion microorganisms inside the gut. These organisms which are helpful may be bacteria, viruses, non-pathogenic fungi, and these play a very important role in fermentation of the food, digestion, metabolic, immune, and neurological function. And let me be very clear, 95% of the body microbiota is found in the gut. So this is how the microbiota plays a very important role. If you look at the ratio between the human cells and microbiota bacterial cells, it is 1:1. That means if you take the entire body cells in our body, and if you take the gut microbiota, it can be considered as 1:1. However, over 100 times the amount of genomic which is there in the bacteria and archaea content is more than the human genome. That means if we take the human genome and bacterial genome, it is 100 times more in the microbiota. The combination of both human genome and microbiome are called as metagenome. Let's understand what is this microbiome. Microbiome is the total genome content of the microbiota. So let's understand what this microbiota, where is it only in gut? No, it is also present in skin, airway, urogenital tract, eyes, and GI tract. These microorganisms play a crucial role in physiological functions, not only with regard to digestion, respiration, passing urine, and also in the eye. So these are commensals. If you look at the microbiota, there are 1,000 plus different species of bacteria in our gut. Each human requires around 50 to 60 grams of sugar per day to keep this microbiota going. And if you look at the 60% of the stool of human being has both dead or live bacteria. Microbiota is unique to the individual, but at the same time, we do share some of the microbiota among our colleagues, friends, family members within the same geography. Even the identical twins have a different microbiota. As I mentioned, gut is a bioreactor. Here, the food intake occurs, and by the help of this microbiota, fermentation of the food occurs. That means complex food particles are degraded by this bacteria. Once the fermentation occurs, further it is digested with various enzymes and it is absorbed for energy. Hence, gut is considered as an awesome bioreactor in our body. Once the fermentation occurs and energy is extracted, the byproduct is gas. Average per day, each human being produces approximately 2 to 4 liters of gas after the fermentation. And if you look at the total volume of the microbiota in the gut is, by volume it is 1.5 liters, by weight it is 2 to 3 kgs. That means microbiota, although we had not known for many years, now in the past 2 to 3 decades, the research has increased phenomenally. Let's understand whether the microbiota starts after the adult or by birth. Actually, microbiota starts from the day one till death. That means we can consider it as a life course perspective. The development of microbiota is generally believed to begin from the birth and continues throughout the life. After the birth or during the birth, when the child passes through normal vaginal delivery, the bacteria which are present in the vagina of the mother will be considered as one of the first microorganisms which are seeded in the microbiota. Similarly, if the child is born by C-section, the microbiota organisms are less. Also, further depending upon the breastfeeding or artificial feeding, and also what type of diet we take, and also the child is fed, on that basis also microbiota development is directly dependent. Further, various illnesses, diarrhea, medications, especially antibiotics. Even a single dose of antibiotics can change the diversity of microbiota. Vaccination, stress, the way we share the food, environment, and being close and intimate with other person also play a role in the diversity of microbiota. By around 3 years of the child, the composition and the diversity of the microbiota is equivalent to adult. That means in a child who is 3-year-old, their microbiota is equivalent to an adult. The density and the composition of the microbiota are affected by chemical, that is fertilizers in the environment, nutrition, what we eat, environmental toxins, and immunological gradients along the gut play a crucial role in the development of microbiota. This diagram clearly talks about how the microbiota develops over the lifetime. If you look at the top one, prenatal and postnatal, there are hypotheses which says the microbiota starts developing as soon as the placental development occurs. That means mother transmits microbiota to the child through the placenta. This is during the prenatal. During the postnatal, it is through the delivery, through the breastfeeding, and also diet. If you look at the down, here the contribution is from the mother initially, later the food we take, and also in the environmental toxins, and also the food we take from the environment also play a role in the development of microbiota. Now, if you look at the diet, the breast milk or artificial milk, what are the solid foods, home, sharing of food, vegetarian, non-vegetarian, individual diet, packed food, or else ready food, and also whether we are using the special diet, all these play a role in the development of microbiota. Further, if you look at the development of various illnesses, especially mental disorder, you can see in the second decade of life, that is, you can see if there is a disruption in the microbiota, there are studies which are indicating the development of depression, anxiety, eating disorder, drug abuse, bipolar disorder, schizophrenia, autism, Alzheimer's dementia, and Parkinsonism also been implicated. But however, with regard to brain function, memory, cognition, impulsivity, language, and social interactions play a role. Further, if you look at the gut, the development and the microbiota continuously occurs over a period of time. This microbiota also is essential for the development of immunity, regulation, and also filtering and keeping the immunity active. Further, as you see, the influential factors such as when the child was born, whether it is a C-section or vaginal delivery, what type of food is given, and whether the child was reared in the rural area, urban area, all those play a role. How often the antibiotics is given, and also if you look at the brain, and you can see various synaptic neurogenesis occurs over a period of time based upon the microbiota. This has been implicated and also been proved in the rodent lab studies. Now let's understand what are the functions of this microbiota. First and the foremost, microbiota plays a first line of defense. Hence, it maintains the integrity of the mucosal barrier in the gut. Further, it also helps in fermentation of the food and digestion of the food to extract energy. It also helps in providing various nutrients such as short-chain fatty acids, vitamins, metabolites, neuroactive substances, gut hormones, and others. Food intake and craving to a particular person also dependent upon microbiota. And as I mentioned earlier, immunity. That means microbiota plays a crucial role in induction, training, modulating, regulating not only the local immunity in the gut, but also the systemic innate and adaptive immune response. That means GIT plays a very crucial role in development and training of the immunity. The interaction between the commensal microbiota and the mucosal immune system is very essential for proper immune function. Any change can also trigger autoimmune diseases in human being. Further, microbiota and brain axis is well known and there are studies pouring in. Further, gut and pulmonary axis, gut and cardio axis, gut and renal axis are also play very crucial role. Further, microbiota also plays a crucial role in learning, memory, and cognition through neurogenesis, neuroplasticity, neuroreceptor plasticity, and neurohormonal plasticity. Microbiota also been implicated how the drugs are metabolized and development of side effects in human being. So after discussing till now about the microbiota, let's understand certain definition and certain words which are there in this field. Microbiota. What does it mean? It is the community of microorganisms in the gut. They include bacteria, fungi, eukarya, and also archaea and viruses, and also few of non-pathogenic fungi. What is microbiome? The collective genome of the microbiota in the gut is called as microbiome. That means it is the genetic material of the microbiota. What is this microbiota diversity? It means it is a measure of how many different species and dependent on the diversity indices, how evenly distributed they are in the community. Lower diversity means that is dysbiosis. More the microbiota diversity means the person is healthy. So let's understand what is this dysbiosis. The opposite of microbiota is dysbiosis. That is altered gut bacterial composition, also considered as microbial imbalance or low diversity, has been associated with the pathogenesis of autoimmune disease and obesity in human being. That means dysbiosis means there is destruction or alteration or decreased diversity of microbiota. What is this colonocytes? They are epithelial cells of colon. They play a very crucial role in defense, immunity, and also in digestion of the food and production of various neurohormones and various other metabolites. Short chain fatty acids. Here fatty acids with the 2 to 6 carbon atoms that are produced by bacterial fermentation of the dietary fibers. Without microbiota, human being will not be able to use this fat and also degrade them into short chainy fatty acids, which are useful for us. That means microbiota plays a crucial role in production of short chain fatty acids. Let's understand some of the important terms. Probiotic. Probiotic are healthy or friendly bacteria that live inside our gut and support our health by converting fiber into compounds that have benefits for our health. Probiotics means these are friendly bacteria. Now there is one more term called as prebiotic. These are group of nutrients, mainly fiber, that feed the beneficial bacteria in your gut. That means probiotics are bacteria. These bacteria needs a medium. That medium is fiber, that is prebiotics. Now there is another important term, postbiotics. These are bioactive compounds the probiotic bacteria produce when they consume fiber. That means probiotic is a friendly bacteria, prebiotic is a fiber. When bacteria acts on this fiber, produces postbiotics. And further, to add complexity, there is something called as synbiotics, which are a combination of probiotics and prebiotics, in which prebiotics are used to supplement, that is providing a source of fermentable fiber and enhance the viability of probiotic and postbiotics. So these are the terms which you need to remember. But however, you need to know about probiotic, prebiotic, and postbiotics. Let's understand the role of microbiota in lab. As I mentioned, in the past 2 to 3 decades, there has been various research occurring in this area. And this research play a crucial role to understand the role of microbiota in our human being. Let's look into that. The studies conducted to identify and examine the microbiota have been used different methodology. They play a very important role in understanding the complementary and also the intervention methods used in microbiota and how they are useful. The first and the foremost is germ-free rodents. I'll be talking about that shortly. Further, antibiotic-induced depletion. Here you give antibiotics and destroy the microbiota and then see by introducing certain bacteria whether it is helpful or not. Prebiotic and probiotic supplementation, whether is it helpful for human or not. Further, gastrointestinal infection and also fecal microbiota transplantation. These are the simple methodology have been used to understand the microbiota. The very important is germ-free rodents. Let's look into these germ-free rodents, are also called as mice. These are not but naturally colonized by microorganisms. Here, when the rodent or the mice is born, they are born not through a natural method. They have been removed by a cesarean section so that they are germ-free. And they are kept in a sterile environment. Sterile environment like this. That means the rodents are kept separately and they are handled in a sterile environment. And if you do any kind of culturing and to look for bacteria, there will be no bacteria yield on culture. That means they are completely germ-free. Now, by introducing various bacteria and see how these rodents grow or not. And these germ-free animals play a very crucial role in understanding whether microbiota is helpful or not. There are various studies have been done. The studies such as where a hyperactive rodent stool is utilized and introduced into the mice which is germ-free, and see whether after introduction of the gut microbiota of a mice which is very hyperactive, after a few days you can see germ-free mice also becomes hyperactive. Similarly, if you introduce a mice which is very silent and withdrawn, if you use those stool or the microbiota from that gut of the mice to a germ-free mice, and you can see that within a few days the germ-free rodent becomes silent. So these kinds of studies are occurring day in and day out. Now, they have studied these germ-free mice after the microbiota has been introduced. They have found there are differences with regard to germ-free mice and also a normally fed mice. In germ-free mice, they have found that their gut is leaky. That means the pathogens can grow easily into various parts of the body. That means it is highly vulnerable for infection. They do not have good immune system. Their immune system is very poor and very weak. They do not able to digest. They are generally weak. Nutritional deficiency is characteristically seen, and also poor functionality is seen in these mice. There is decreased learning, poor motivation, poor memory, and also cognitive functions are very less. Further, lack of socialization can be seen in germ-free mice. And there are significant changes seen in these germ-free mice, such as decreased neurogenesis, decreased microglial activity, decreased synaptic and neuroplasticity, decreased myelination, and also myelin sheath, and also decreased brain-derived neurofactor. Blood-brain permeability is very high, gut permeability is very high, and also decreased hippocampus and amygdala volume, which indicates there is a decreased memory, decreased learning, and also decreased neuroplasticity in these areas. Let's understand what are the research on microbiota. With the advent of culture independent approach, that means you need not have a culture to understand these bacteria. Culture independent approaches to identify the microbiota targeting the bacteria, that is the bacteria's 16S ribosomal RNA. This RNA gene sequencing helped us to identify. In this 16S ribosomal RNA gene is present in all bacteria and archaea and contains 9 highly variable regions, that is V1 to V9, which allows us to identify the species and also able to know what is the strain which it belongs to. The utilization of this shorter read length can also add to the error. Hence, there was a further refinement in the technology. To understand this, the composition and diversity of microbiota may be provided by whole genome shotgun metagenomics. Due to the higher resolution and sensitivity of this technique has been utilized. It means from 16S ribosomal gene and further refinement into whole gene shotgun metagenomics have been utilized to understand the microbiota. And various important research projects have occurred, such as MetaHit, Human Microgen project also has been considered. Further, Microbiome project also are very important to understand the microbiota. The results from these projects, the compiled data from these projects identified 2,172 species were isolated from human beings and classified into 12 different phyla. In that, 386 species are strictly anaerobic in nature. So, these have been identified and subtyped. Further, an extensive catalogue of the functional capacity of the human gut microbiome was recently obtained, where 99,87,896 genes were identified through a combination of 249 newly sequenced, and you can find 10,018 published samples. The study identified the presence of country-specific microbial signatures, suggesting that gut microbiota composition, even if it is shaped by the environment factors such as diet and also the host genetic factor. For example, if you want to study the microbiota in a geographically well-defined location, there may be sharing of microbiota. That means you can see the microbial signature from specific place depending upon what food they eat, what is the environment, how much fertilizer is used, how much organic fertilizers is used. All this plays a role in the development of microbiota and also maintenance over a period of lifetime. Yes, microbiota is not constant. They're continuously changing. Gut microbiota is continuously changing in the gut environment. Ecosystem of the bacteria in the human gut operates dynamically. It depends directly upon the lifestyle. What is the physiological state of the person? What food intake, stress, smoking, alcohol intake, antibiotic intake, genetic vulnerability, from which place he is coming, geography, age, gender, resilience, the type of birth, and also various other factors plays a role in this microbiota, and it changes every moment and every second. Further, the interaction of the microbiota and diet also plays a role in the development of the microbiota. Further, this microbiota ferments the food, and that smaller part of the particles will be considered in the protein and the development of genes. And these genes and also those broken down food particles can be considered as a metabolite of microbiota. Let's understand microbiota histologically. The mucus is present throughout the gut, that is GI tract, and it is thickest in the colon, where it is crucial in mediating the host microbiota relationship. If you look at the colon mucus, it is divided into two layers. A dense, impermeable, sterile inner layer, a loose outer coating which is penetrable by bacteria, and energy source. Here, the preferential binding site for commensal bacteria occurs. Let me repeat. The mucosal of the colon has two important layers. A dense, impermeable inner layer, which is sterile. The outer layer is loose and it is highly penetrable. And here, the commensal bacteria are attached and the energy source starts occurring here. If you look at this diagram, you can see the gut where the microbes are there, where microbiota. From there, after you can see that clearly the genes and proteins are formed after the fermentation and digestion, and further it is produced is the metabolite. You can see the bottom-up approach and also top-down approach. First, as I mentioned, that is the gut microbiota, where you can see in the bottom-up approach various bacteria are there and also various fungi and various other microorganisms. This will degrade the food or ferment the food further, and there they produce various genes and proteins, and further it is degraded into various metabolites. In the top you can see the healthy gut, that is, there is no inflammation and you can see the thick mucus. But however, as you go down here, the mucus is reduced. That means the mucus is not there and the gut has been compromised. Here, the cells, pathogens can easily enter into the gut, that is basically into the wall of the gut, and producing inflammation and causing various illnesses in the host. Further, as I mentioned earlier, there are various metabolites produced from the microbiota. The gut microbiota provides essential capacities for the fermentation of non-digestible substrates like dietary fiber and endogenous intestinal mucus. That means some of the fibers we do not have a capacity to digest. Microbiota helps them. This fermentation by the microbiota supports the growth of specialist microbes that produces short chain fatty acids, metabolites, and other gases. Here we are very interested in short chain fatty acids. These short chain fatty acids is very important nutrient for our body. The major part of the short chain fatty acids are butyrate, acetate, propionate, and various other metabolites. Here we will discuss each one of these: butyrate, acetate, and propionate. Butyrate. It is the main energy source for human colonocytes. Can induce apoptosis of colon cancer cells. That means the butyrate plays a very crucial role in protecting against cancer. Further, it also plays a crucial role in the intestinal gluconeogenesis. That means in extracting energy. It also plays a crucial role of hemostasis through beta oxidation, generating a state of hypoxia that maintains oxygen balance in the gut and preventing the gut microbial dysbiosis. That means butyrate also indirectly helps microbiota's environment. Let's understand about the acetate. Here, it is the most abundant short chain fatty acid, an essential metabolite for the growth of other bacteria. Here, this acetate helps in the growth of other bacteria. It is used in the cholesterol metabolism and lipogenesis, and that means 15 to 20% of the energy is produced here. May play a role in the central appetite regulation. The acetate, once you take the food, and food is fermented in the microbiota, and further acetate is formed. This acetate may play a role in giving feedback to the hypothalamus: yes, this person has taken food and the appetite is suppressed. Now let's understand about propionate. Propionate, once it is formed in the gut, it is transferred to the liver, where it regulates gluconeogenesis and satisfies the signaling, satiety signaling, through interaction with the gut fatty acid receptors. And butyrate and propionate control the gut hormones. Further, other products are also formed in the gut, such as trimethylamine. This is a toxic substance which helps in development of atherosclerosis. That means we need to decrease trimethylamine formation. If the atherosclerosis is developed, as you know, the artery pliability becomes very less and hypertension develops. Similarly, there is indole propionic acid development. This also causes diabetes too. That means there are some metabolite which should not occur. Whenever there is a dysbiosis or the microbiota changes rapidly, and because of the food we take or fatty foods we take, or else the cooked food, or it may be ready food which is available on the shelf if we take, may produce this trimethylamine or indole propionic acid. They play an important role in the development of these illnesses. There are various other metabolites are being studied, such as gut hormones, neurohormones, neuroactive substances, liposaccharide, polysaccharide, peptides, enzymes, glycoprotein, microbiome, that is, nervous system of the gut, pro-inflammatory cytokines, ghrelin, and 5-HT plays a crucial role, and other metabolites. At this point of time, we know few of these. There are many substances we are unable to code at this point of time because research is currently in progression. Let's understand about dysbiotic. As I mentioned earlier, dysbiotic means decreased microbiota, or else altered microbiota, decreased diversity of microbiota, or else decreased density of microbiota. Here, these altered microbiota is not helpful for human being. This dysbiotic microbiota might be the one which fails to provide the host with the full complement of beneficial properties. That means the products which are formed after fermentation and digestion is harmful to our body rather than providing energy. Here there may be a reduced diversity of the microbiota, decreased number of commensals or helpful bacteria, and there may be pathogenic microbiota. That means harmful bacteria can start growing. And a plethora of diseases have been implicated to dysbiotic, from development of neurodevelopmental disorder to psychiatric disorders. If you look at this diagram, on the right side you'll be able to see there is a green color which talks about the helpful microbiota, where there is a dietary intake is there, probiotic intake is there, increased short chain fatty acid, increased antioxidant, decreased indoxyl sulfate, which is kidney toxin, further improved lipid metabolism, low gut inflammation, insulin sensitivity, and reduced risk for infection. That means on one hand, you can see the health. On the other hand, you can see there is a disease. What are those diseases? At the end, you can see there is altered pH in the gut, which leads to excessive protein consumption and also excessive sugar intake, excessive saturated fatty intake, proton inhibitors, antibiotics, which play a crucial role, and short chain fatty acids are decreased, and also various other changes occurs, which leads to various illnesses like hypertension, diabetes, cognitive decline, and neurodevelopmental disorder. So that means the early or preliminary studies indicates microbiota plays a crucial role in various illnesses, not only from the brain, it also in the pulmonary, and also with regard to systemic hypertension, diabetes, endocrinal system, renal system, colon system. That means microbiota cuts across various departments or else various parts of medical fraternity. We will focus on currently on microbiota-gut-brain axis. Here, one of the important understanding you need to understand. Trust your gut feeling. What is this gut feeling? As you know, GIT has maximum number of fibers with regard to neurons. Hence, it is considered as a second brain. The first brain is the brain which has neurons, and the second brain is GIT. That means here there are maximum number of neurons, maximum neurohormonal production occurs here. What is this microbiota-gut-brain axis? That is, this axis is a dynamic bidirectional communication between the gut microbiota, gut and the brain through neural, endocrine, and human pathway. Let me give you an example. Look at this figure. In this figure, you can see it is a bidirectional communication between the brain and the gut. That means brain will influence the gut. At the same time, gut also will influence the brain. Here, how does it influence? The pathway are immune system, neuroendocrine system, nervous system, circulatory system, and vagus nerve. Through this, both brain and the gut communicate each other. Further, there are various neurohormonal secretion, peptide secretion, and also neuroactive substances. Microbiota-derived products are developed, and also various metabolites are occurs here. Along with this gut also play a crucial role in altered receptor activity in the brain, altered neurotransmission in the brain, and also metabolite entry which occurs in the brain. With this, it causes neuronal plasticity, learning, memory, and motivation. Further, if there is any altered microbiota or else dysbiosis, causes various neurodegenerative disorders such as multiple sclerosis, Parkinsonism, Alzheimer's dementia, brain tumor, ischemic stroke, hemorrhagic stroke. Further, various neuropsychiatric illnesses such as autism spectrum disorders, depression, anxiety, stress, and schizophrenia. That means microbiota plays a role in development of these illnesses. It may not be the direct cause, but it may precipitate these illnesses in genetically vulnerable individuals. But however, the research is ongoing. Further, the gut microbiota has extensive reciprocal connection with the human brain through microbial metabolites, vagus nerve, spinal nerves, hormone, and immunological signaling, collectively forming microbiome-gut-brain axis. Earlier it was microbiota, now I am mentioning microbiome-gut-brain axis. That means the neurons from the brain connects the gut, and from the gut it also connects the brain through vagus nerve, spinal nerve, and also through various neurohormones and immunological ways it communicates. Here the diagram is very clear. It talks about involvement of circulatory system in microbiota-gut-brain axis. Here you can see various metabolites are released into the artery, and those artery will carry blood directly to the brain. And that means from the brain, it communicates the gut, and the gut communicates not only through neurons, it also communicates through circulatory system. Further, you need to understand there are two important barriers. One is blood-brain barrier and gut-blood barrier. So these two barriers play a very crucial role in defending the pathogenics and also allowing only the required nutrients to pass through. But however, whenever there is an altered gut microbiota, these both blood-brain barrier and gut-blood barrier permeability increases. That may leads to various neurodegenerative disorders or psychiatric disorders. Now let's understand the regulators of microbiota-gut-brain axis. What are those regulators? Here you can see the important regulators. First and the foremost, the lifestyle, exercise, diet, sex differences, that is between male and female there is a difference, genetic vulnerability, epigenetics, social interaction, environment, fertilization, or else fertilizers used, insecticides, pesticides, or else pollution, environmental pollution, stress, health, medication, especially antibiotics, and the way the delivery method has occurred, whether it is vaginal delivery or the cesarean section. All these play a very important gut-brain axis regulator. That means these do play an important role in regulation of microbiota. Further, gut-brain axis, as I mentioned, the gut, although it is a largest digestive system, but also a largest human system, endocrine organ, and enteric nervous system. That means nervous system is, it is a second brain. The brain is connected with the gut through these bidirectional pathway via above routes, that is circulatory, immune, endocrine, and enteric nervous system. Let's understand the microbiome gut-brain axis. You can see here the brain is connected to gut through not only through the spine but also the vagus nerve. But how does it play a role? Through HPA axis, that is hypothalamic pituitary axis. Through it may act directly on the thyroid, on the adrenal gland, gonads, and various other places. You can see there is afferent nerves and also efferent nerves. Further, immune system, B cells, T cells, cytokines also play a role in immunity, which connects to the brain. Further, these immunity and also neurohormonals formed like maybe serotonin, dopamine, norepinephrine also play a role in the connection between the brain and the gut. First, let's understand about the neural pathway. The gut microbiota influences the synthesis and the secretion of various neurotransmitters, from serotonin to GABA. The gut signals can activate the enteric nervous system and primary efferents as well as transmit the message to the brain through the vagus nerve and the sympathetic nervous system. That means brain is connected to the gut through vagus nerve. Both afferent and efferent system play a crucial role. Not only that, it also plays a role in the development and production of various neurohormones. Very important is serotonin. Let's understand the intestinal tryptophan metabolism and formation. There are three important metabolism where the serotonin is formed. One is direct transformation, second one is kynurenine pathway, third one is formation of serotonin using enterochromaffin cells. So these three important pathways helps in formation of serotonin in the gut, and gut is the largest organ which produces the maximum serotonin in our body. You can see all the three important. One is the formation of the gut microbiota forming serotonin through the direct metabolism of the microbiota. Second one is through kynurenine pathway and through the enterochromaffin cells. That means the gut produces maximum serotonin. Further, the second pathway is endocrine pathway. Even the endocrine pathway is where the gut microbiota impacts the concentration and function of the neuropeptides, and that includes neuropeptides, gut peptides, OT, opioid peptides, neurohormones such as melatonin, communicating with the brain through the neuroendocrine pathway, including HPA axis, thalamus, hypothalamus, pituitary, and other peripheral glands are involved. That means endocrine plays a crucial role in the connection between the brain and the gut. You can see all those important axes. First and the foremost is the HPG axis, that is hypothalamic pituitary axis. Here they are connected with the gonads, that is estradiol and testosterone, where FSH and LH plays a crucial role. On the other end, there is HPT axis. Here hypothalamic pituitary thyroid. Further, you can see hypothalamic pituitary adrenaline axis and HN axis. Here the brain connects the gland and also the gut through pituitary. From pituitary, various endocrine systems are connected. These endocrine systems plays a crucial role in digestion, development of nervous system, and also resilience. So you can see the whole picture you can be captured in this simple diagram. Further, immune pathway. The third, the gut microbiota regulates the function of TGs and TLRs in the gut barrier and blood-brain barrier. They adjust the differentiation of lymphocytes and impacts the brain via the immune pathway. Additionally, the bioactive products which are produced by microbiota also influences the microbiota-gut-brain axis. Here, basically cytokines, interleukins, and various other metabolites which are produced by microbiota plays a crucial role. This is how it impacts. You can see here the blood-brain barrier. Similarly, the gut-blood barrier. Further, you can see gut microbiota, and you can see the nervous system, vagus nerve connecting the brain to the gut. Further, you can see sympathetic nervous system connecting brain to the gut, and also the immunity system on both the side. On the left side, you can see neuroinflammation. On the right side, neurogenesis and neuroprotection. Both are seen in this diagram. Let's take each one of them. First, neurogenesis and neuroprotection. As I mentioned earlier, there is a blood-brain barrier, and from the brain, hypothalamus, pituitary, and peripheral gonads or glands are connected. From here, the hormones are secreted, and further, the impact on the gut can be seen. Further, not only that, through vagus nerve, it is connected to the gut through sympathetic and parasympathetic. Here, the essential is parasympathetic. Further, from the gut, various cytokines and also immune-mediated response, which are essential for the neurogenesis and neuroprotection. And you can see various neuropeptides, gut peptides, opioid peptides, melatonin, and various which are very essential for neuroprotection and neurogenesis occurs here. Similarly, if there is any change in the microbiota which causes neuroinflammation and neurodegeneration, here you can see the overactive of plasma and basically the immunity, and also if there is the hyperactive sympathetic system which causes various unwanted metabolites which increase the permeability of the gut and also the blood-brain barrier. And that leads to various important illnesses like neurodegenerative disorders and also cause of various psychiatric disorders. Let's understand what are the important metabolites which are commonly studied in our research. There are three important, but here we will be focusing only on short-chain fatty acids, which are produced by the bacteria by the fermentation of the fibers, and also there are bile acids which are produced from the liver and goes into the gut. From there it recycles. Further, tryptophan metabolites which also play very important. These are the important metabolites which are required at this point of time. Why I'm saying at this point of time? Because research is occurring and further research will lead to recognition of various other metabolites. This is the serotonin formation in the gut, and as I mentioned earlier, there are three important ways, and the serotonin are formed in all the three ways. And let's discuss about the management of dysbiotic. If microbiota has been altered or the diversity has been decreased, how to manage this? What are the important ways we can do it? Depending upon the type and the stages of disease, following important method is approached. One is microbiome modulator. Here, the antimicrobials diet and prebiotic and probiotic mostly aimed at the changing the composition of the host microbiota is done. Further, microbial-based solution. Here to replace some of the defective microbes and their associated benefits that have been done. It's very difficult to understand, but however, let me give you simple examples which can be understood. First and the foremost, fecal microbiota transplantation. From a healthy person, you take the microbiota and transplant into a deceased person. The fecal microbial transplantation of a solution of a fecal matter from a donor into the intestinal tract of a recipient in order to directly change the recipient microbial composition and confer the health benefit. This is FMT. FMT has been studied in a major way in a Clostridium difficile infection, inflammatory bowel disease, irritable bowel syndrome, obesity, and metabolic syndrome. But however, the study are occurring, but there is no robust study to say yes, it is helpful. But however, in some of the study it has been found to be very helpful. First and the foremost, before you do fecal microbiota transplantation, you need to select a donor. A donor without a family history of autoimmune disease, metabolic disease, malignant disease, and screen for any important potential pathogens. Once you have screened, the fecus has to be collected, prepared, mix it with the water or a normal saline, followed by filtration, and steps to remove particulate matter has to be done. Once you've done that, the mixture can be administered through a nasogastric tube or enterogastric tube. Either it can be directly placed into the gastrium, but however, if there is hydrochloric acid is very high, it may destroy it. Or else you can place it in the jejunum, intestine, or a small or large intestine. Or else you can directly place it through colonoscopy or through retention enema. So these are the various methodology adopted in fecal microbiota transplantation. The second important is antibiotic. Here, already the microbiota is altered. You need to destroy or else to change the pathogenic microbiota which is there in a deceased individual. Here, the antibiotic plays an important role in the changing the composition of the gut bacteria which are pathogenic in nature, which influences the immune system. Hence, we need to give antibiotic and destroy those pathogenic bacteria. Hence, here antibiotic plays a therapeutic role. However, the antibiotic has been used in gliomas. For example, antibiotics are essential for preventing or treating the life-threatening infections in patients suffering from microbial-induced immune suppression. Here, the studies have demonstrated the broad-spectrum antibiotics has been useful. But however, a normal people using antibiotic can destroy the microbiota. Further, probiotic using. Probiotics are those which are friendly bacteria. These are all those compounds or components of food, products and supplement that we consume daily. It may be yogurt, buttermilk, kimchi, and various other products which has friendly bacteria. These are increasingly becoming designer probiotics and have been introduced that are genetically engineered to maximize the beneficial effect of selected bacteria and often included in the more traditional pharmaceutical routes. But however, we do not know the long-term consequences of these probiotics. Further, body bacteria and lactobacillus, which promotes GABA level and expression of neurotropic factors, including reduced occurrence of seizure episodes in patients with drug-resistant epilepsy has been found. Further, there is a wide variety and variability in the composition and stability and authenticity of these probiotics. Hence, we do not know in the long run how it will be helpful. What should be the dosage? What should be the duration? What is the specific strain to be used in probiotic? We don't know. But however, probiotics are flooding the market at this point of time. We do not know the long-term impact of this. Probiotics do not permanently change the gut bacteria. That means you need to take on daily basis. That means frequent consumption of probiotic is required. Here, probiotic do not change the microbiota of the gut. This is how the example which can be given. Helicobacteria and Escherichia coli. These, if they are there, E. coli are there in abundant, which causes various neurodegenerative disorder. Healthy bacteria, which are bifidobacterium or else lactobacillus. These help in maintaining the health and homeostasis. Further, diet and prebiotics also play a role. Diet, though not necessarily a therapeutic treatment, also play a significant role, especially in accumulating evidence indicate that intensive changes to dietary regime can rapidly alter the gut microbiota profile. And a specific dietary regimen, which are considered as a prebiotic, which are fiber in nature, can be given to the human so that the microbiota can be altered. This requires long-term intake of prebiotics. At the same time, you need to change the diet if you are using high saturated oil, also various other oily foods you are taking. Diet and prebiotic have been found to be useful in epilepsy patients. The ketogenic diets composed of high fat, low carbohydrate and adequate proteins were found to decrease the number of seizure episodes by increasing GABA and decreasing glutamate. Let's look into the impact of microbiota in psychiatric disorders. There are various studies have been done, but however, these studies are done in rodents. Let's look into the recent systematic review published in Psychosomatic Medicine by Long and his colleagues. They did a systematic review which included 69 studies which were conducted on various psychiatric disorders such as autism spectrum disorder, depression, eating disorder, schizophrenia, bipolar disorder, ADHD, generalized anxiety disorder, and PTSD. In the review, the results came, they were stunning. That study clearly said that short chain fatty acid producing bacteria were less in patients with psychiatric disorder. But however, these are initial studies. There are no studies done on human beings robustly or systematically. RCT studies have not been done. Hence, this systematic review concluded by telling that the study of intestinal microbacteria in psychiatric disorder is characterized by minimal cohesion and few reliable replications can be seen. But however, there were a lot of methodological issues which prevail in microbiota studies. Small sample size, differences in patient selection, diagnosis, failure to exclude or account for confounding factors, and also shortcoming in bioinformatic analysis. There are two important confounding factors. One is demographic variables, age, gender, ethnicity, geography, and BMI. They need to be controlled. Further, environmental factors such as diet, medications, lifestyle factors also need to be controlled. That means the studies which are done at this point of time have a poor methodology and lot of confounding factors. There are various ongoing trials are occurring. These have been published in Longsulo et al. And you can find these. Still, the studies are ongoing. That means whatever we know about microbiota are preliminary in nature. Further, these preliminary studies demonstrate that intestinal microbes may be intimately involved in the progression of disease of the central nervous system, including mood disorder and psychiatric disorders. Gut microbes regulate inflammatory situations, play a role in the balancing the concentration of neurotransmitters, and provide beneficial effects against neurodegeneration. These are the preliminary findings. What is the implication? Implication of microbiota-gut-brain axis in day-to-day practice. As we know, gut health is a key to overall health. So what should we do at this point of time? At this point of time, the results are very preliminary in nature. How it should be translated? First and the foremost, here you need to avoid unnecessary antibiotics. We need to educate the public at large not to gulp the antibiotics as and when required. That needs to be prescribed. First, that is very essential. Second, diet management in psychiatric disorder becomes very crucial. Whenever we give antibiotics, we need to avoid that. At the same time, whenever we give antipsychotics, mood Regulators, we need to regulate the diet also. Here, we need to educate the patients to reduce saturated fats, eliminate trans fats, eat food rich in omega-3 fatty acids, high fiber diet, lose weight, avoid alcohol, drugs, and stop smoking needs to be done. Further, lifestyle also plays a crucial role. Regular sleep, stress management, daily exercise, meditation, and yoga also play very important role. That means we have a role to play, especially with regard to the psychosocial management. That is diet management, and also the exercise, sleep, all these play a very essential role in the management of microbiota. So, during the acute stage of illness, patient and family members will be receptive. Hence, you need to talk about lifestyle management. So, lifestyle modification play a role in the formation of good microbiota. Hence, you need to utilize this opportunity with the patient and the family member. You need to advocate about the diet and lifestyle management. If there is obesity, you need to give cognitive behavioral therapy referral to reduce obesity. And if there is anger management, stress management need to be done. In the research of microbiota, there are a lot of unanswered questions, such as the same microbe could do harm and also benefit in an individual depending upon the genetic vulnerability. So we do not know about the genetic vulnerability and resilience of this individual. The second, a single bacteria may have various strains. We do not know what is the effect of each strain on the given individual based on the genetic vulnerability. Third, microbial profile varies widely within a given individual based upon the lifestyle. That means it is highly dynamic in nature. So, what is the role of the environment and the weather also on the microbiota, we do not know. Further, microbial profile varies widely between the individuals within a given community. So, it is very difficult to know whether how much the microbiota is playing a role in the development of disease. Further, microbial profile varies widely between individuals across community and also across continents. That means the vulnerability and interaction with the microbiota leading to illness needs to be studied systematically by removing these confounding factors. To conclude, my dear friends, the evolution of microbiome field has enabled significant step closer to the understanding of CNS disease and treatment. Through untangling the intricate networks of human system, gut microbiota and microbiota-gut-brain axis play a crucial role. It is the first step in understanding about the mystery of the CNS disorder and psychiatric disorders. However, there are many challenges, at the same time opportunity in understanding the role of microbiota in our human being. At this point of time, microbiota research is still in infancy stage. Despite the limitation of what it is known, the gut-brain axis is a complex but exciting area of investigating the role of microbiota in development of neurological disorder, psychiatric disorder, and neurodegenerative disorders. The interaction between microbiota, microbes, gut needs to be studied in a systematic manner. Still a long way to go, but we have an opening or a window to understand the role of microbiota in human beings. Thank you very much for giving your valuable time. Stay safe.